Welcome to Chem 313, lecture number 19. Today's topic is going to be DNA replication. And many of you may have seen this already in other classes, and so it should be largely review for you. And probably I'm not going to expect you to know it in much more detail than the other classes that you already took would have you know it in. So to review from last time, we talked about nucleic acid structure. And we said that DNA stability is due to two factors. First of all, we have base stacking within the double helix. So you'll remember that we had the diagram that showed the double helix like so. And if you look at it side on like this, you can see that the bases stack up almost perfectly in there. And then we had base pairing. And base pairing is the specific hydrogen bonding between different bases on the opposite strands. So remember, we have two strands in our DNA. It's two molecules. We have, we'll call them strand 1 and strand 2. And they're anti-parallel to one another. That means the 5' end of one lines up with the 3' end of the other. And base pairing is when we have either A on one strand, hydrogen bonding with T on the other strand, or U if it's an RNA. And we have G on one strand pairing with C on the other strand. And notice the way that I drew the base pairs here. A and T base pairs or A and U base pairs always have two hydrogen bonds. G and C base pairs have 3 hydrogen bonds. Now we also talked about so-called secondary structure in RNA. And this is where the RNA, which is usually single-stranded, folds up on itself to optimize base pairing. And the most common type of secondary structure that you see in RNA molecules is something called a hairpin, which is something that looks kind of like this. If that's a 5' end and that's a 3' end, we'll have base pairing in that part of the RNA molecule, but then we always have a few bases that are not paired with anything and they just sort of hang out. The base paired part is called a stem. The non-base paired part is called a loop, and the whole thing is called a hairpin. And we said before that you tend to see hairpins when you have split palindromes. So what's that? That's a sequence that, if you read it in reverse, is complementary to itself. So for example, AU, AU, AC, and then we have a few that don't necessarily base pair with anything. We'll just randomly say AAGU. And then we have another segment that is complementary to what came before it, but in the opposite direction. G, U, A, U, A, U. And so when this is folding up in solution, we get base pairing interactions that look like this. Didn't leave myself enough room there. There we go. And so you always are going to end up with a few bases in the middle that aren't base paired with anything, but the other ones base pair with one another in the stem of our RNA hairpin. Now, we said before that sometimes RNA structures are not perfect. Not everything is going to form a correct Watson-Crick, as we say, base pair. So, for example, one of your bases may be mismatched. Instead of an A to U, let's say you have an A to C. And so in those cases, what ends up happening is you have an imperfect base pairing in your RNA hairpin, which we indicate with the dot. And sometimes that'll introduce a little bulge or a kink in your RNA molecule, which is why RNAs have such diverse structures. The other topic that we briefly introduced with respect to DNA is the notion of fidelity. And we said that the fidelity of DNA refers to the ability to be copied repeatedly with few, if any, errors. And we said that while DNA stability is partially due to base pairing, although base stacking is actually a bigger factor, we said that the real importance of base pairing is in fidelity. Fidelity of DNA is due almost entirely to base pairing. And we're going to see how and why that is right now when we talk about DNA replication. All right, DNA replication is the process of making copies of DNA so that cells can divide. Whenever a cell divides into two cells, all of the DNA in that cell must be copied because every cell in a multicellular organism or in single-celled organisms, every cell resulting from cell division has the same genetic material. So that means that if each cell is going to take in the exact same DNA that the parent cell had, then obviously we need to make a copy, because if we just pass DNA from one cell to the next, one cell would have DNA and the other one would not. So what ends up happening is a complete copy of all DNA molecules is made prior to cell division. And before we get into the nuts and bolts of how this happens, it's worth pointing out that this is an enzyme catalyzed process, and it's quite complicated. We're going to give you guys the easy version of it. It's going to consist of many steps, and it's also worth mentioning that massive amounts of ATP are needed in order to drive this process. So we're not going to get into which steps require ATP and why, but it's a massively ATP-dependent process. Cells cannot divide without energy. All right, so there are a few things that the cell must have on hand before it can replicate DNA, all right? So what we're going to do is we're going to give you guys how this works in bacteria. We're talking about bacterial replication here. In eukaryotes, there are more factors than just these, but these are the critical factors for bacterial DNA replication. So what do we need? Well, first of all, we need pre-existing DNA. So that may sound like a no-brainer, it's obvious, but the fact is you can't simply make DNA from nothing. If a cell had the machinery to make DNA, it would probably just put together random bases and you would end up with a random genome, which is useless. So we need to have a pre-existing DNA that functions as a template. Second, you need to have a mixture of all 4 deoxynucleotide triphosphates. So in other words, just our plain nucleotides, not polymerized yet, ATP and magnesium. All right, the deoxy NTPs are our reactants. So the actual reaction that we are going to be catalyzing is this: deoxy NTP plus DNA with N nucleotides in it are going to react together to give us DNA extended by 1 nucleotide. And in the process of doing this, we release PPI, which is pyrophosphate. Pyrophosphate is this ion right here. We mentioned it before when we were doing metabolism. And pyrophosphate, I didn't mention this earlier, but pyrophosphate is very readily hydrolyzed by water to give you 2 phosphate ions. And that reaction, hydrolysis of pyrophosphate, is very, very favorable. Its delta G is much less than 0. So this release of pyrophosphate is what allows the DNA to polymerize energetically. You don't have to use ATP to get this process to work. Now, I know I just said ATP is required. ATP is required for different steps, not the actual polymerization reaction. All right, next thing you need is an RNA primer. So the RNA primer is something that is made as it is needed. We'll talk about what that is and what it's for when we get started on the whole process. And then lastly, you need the DNA polymerase complex. This is your catalyst. So it's a multiple enzyme complex, and each enzyme in it has a different function. So the individual enzymes of the DNA polymerase complex, and this is not a complete list, but it's worth knowing the ones that we have. We have a helicase. We have a so-called beta clamp, which isn't an enzyme, but it's an important protein. We have got DNA polymerase 3, which is abbreviated POL lowercase 3. DNA polymerase 1, primase, and DNA ligase. There are some other accessory proteins in the complex along with those, but those are the critical ones. So those are the critical enzymes that we actually need for replication in bacteria. And what they actually do, we'll talk about in a moment. Now, one thing that you should know, if you don't already know, is that in bacteria, essentially all bacteria have a single circular chromosome, unlike eukaryotes. So all of their DNA is circular. There's no ends to it, in other words. And that ends up being pretty important because copying a circular piece of DNA is, as we're going to see, it's actually simpler than trying to copy a piece of linear DNA like we have. So our DNA replication has to start at this site on the genome called a replication origin. And the replication origin is just a sequence of DNA where the polymerase complex initially binds. And then what ends up happening is, starting at that replication origin, as you synthesize a new DNA, the newly synthesized DNA just sort of peels away from the old DNA, and before you know it, you've got 2 circles of DNA. So for this to work, it has to happen in a bidirectional fashion. So that means that we have to be synthesizing DNA in both directions at once. And this spot where the new DNA grows and splits off from the original DNA is called a replication fork. And in a bacterial chromosome, you have two replication forks, and replication occurs at the forks. So in other words, in bacteria, we need two polymerase complexes to complete the entire replication cycle. So I'm not going to show you guys what happens at both of these complexes because it's the same process. We're just going to take a look at what happens at one replication fork. All right, here's the steps in bacterial replication, and I am going to try to narrate and draw along as I go. So our first step involves the use of the helicase. So what does the helicase do? It unwinds the DNA double helix. So that means that if we start off with our double helical DNA, I don't know why I have such a hard time drawing a double helix, but it's really hard for me for some reason. What the helicase actually does is it twists the DNA, or I should say it untwists the DNA to straighten it. It doesn't do it to the whole molecule at a time, of course, just a small piece of it ahead of the replication fork. But as it turns out, base pairing basically requires that our DNA be double helical. If it's straightened, that stresses the base pairs, essentially pulls them apart, and it causes the base pairing to unzip. So the helicase, it works like a little motor. It actually unwinds the DNA mechanically. So this is an ATP-dependent process. And when our base pairs unzip, that's what starts our replication fork. So unzipped base pairs would look something like this. Here's our replication fork. We've got our two strands of DNA. Our helicase would be up here, unwinding the DNA. And shortly behind the helicase is where our base pairs would rupture. And we're left with two separated segments of DNA that aren't base paired, so they're just single-stranded. All right, now to give you guys an idea of what actually happens, I'm going to erase the helicase and I'm not going to show the enzyme complex. We're just going to draw what happens to the DNA itself. So please understand that in order for all this to happen, there needs to be a gigantic enzyme complex clamped around the DNA right at our replication fork. Okay, so an important thing to remember, once again, our DNA strands are anti-parallel. There's the 5' end, there's the 3' end, which means that on the other strand, it's backwards. Okay, so now we have our replication fork. Step 1 is done. Now, what happens at step 2? Step 2 involves the DNA primase, and the primase synthesizes RNA primers. A primer is simply a short segment of RNA in this case, although you can also have DNA primers in different situations, but in replication it's an RNA molecule that is complementary to the DNA being copied. And it functions as a starting point. Okay, what do I mean it functions as a starting point? Well, it turns out that the DNA polymerase can't start from scratch. It needs sort of starting blocks, if you will. So a DNA polymerase cannot just lay down nucleotides and polymerize them together. It can only add to a pre-existing 3' end. And so what our primer does is it gives us a free 3' end that the DNA polymerase can use as a starting point. So we have two strands, we therefore need two RNA primers. So I'm going to start drawing in different colors to make what I'm drawing stand out a bit better. All right. Let's use green for our RNA primers. That would be the 5' end of the primer. That would be the 3' end. And we need one on the other strand as well. Notice where I'm drawing it. 5' end, 3' end. Great. So the primer is shown in green. All right, next, step 3. We have a free hanging 3' end that DNA polymerase can add on to, or at least use as a starting point. So when we go from step 2 to step 3, the DNA polymerase starts to synthesize DNA in the 5' to 3' direction. When I say in the 5' to 3' direction, I mean on the DNA molecule that is being made, right? So in other words, what's going to happen is it's going to lay down deoxynucleotide triphosphates, and they assemble on the strand that's being copied. By complementary base pairing. And so the 5' end of this new DNA is going to be synthesized, the 3' end is there. And the DNA polymerase works in that direction. That's what I mean by 5' to 3' direction on the strand that's being made. The same thing happens on the other side. And DNA synthesis proceeds. Okay, so all we have to do now is just keep unzipping our DNA, keep synthesizing our new DNA molecules on top of the old ones, and just keep going with this repeatedly. But there's a slight problem with this. Remember what I said. The DNA polymerase complex is this gigantic protein clamped around the replication fork. And it's got the helicase in front, which physically moves. So the entire complex is moving in this direction. And the replication fork splits out behind it. All right, well, let's take a look at this strand of the DNA on top. So on this strand, notice that we're synthesizing DNA in the same direction that we're moving. So on that strand, that seems to be pretty much okay. You have an enzyme that's synthesizing DNA as it goes, and it's just leaving this big trail of DNA behind it as it moves. But on this strand, the bottom strand, that's a problem because notice that the DNA is being synthesized in the opposite direction that the catalyst is moving. So that creates a difficulty. So what ends up happening is DNA synthesis works slightly different on the two strands. So the one at the top is what we call the leading strand. On the leading strand, the DNA is synthesized continuously. Meaning that the enzyme moves along with the rest of the complex and it just lays down a trail of DNA behind it as it moves. On the other strand, we have what's called the lagging strand. On the lagging strand, DNA synthesis is discontinuous. So how does it actually work on the lagging strand? How it actually works is like this. The DNA will be synthesized up to a certain point, and then everything has to stop. And then the primase has to come in, lay down another primer, and then after that, I have to extend my DNA here so I still have space to draw. And then after that, we end up forming another short segment of new DNA. And then we have to lay down another primer, make another section of DNA over and over and over again in these short little bits. And so that may seem to be rather tedious, and it is. I have an animation that I have a link to at the end of the presentation. It shows you how the DNA polymerase complex does it. It's really cool. Anyway, what you're responsible for knowing is that the two strands have DNA being synthesized differently. One strand it's happening continuously, and on the other strand it's happening in little bits. Now, as this whole process proceeds, there is another enzyme called DNA polymerase 1, which sort of lags behind DNA polymerase 3, which is the one that does most of the DNA synthesis. So the DNA polymerase 1 replaces our primers with actual DNA. So that's the function of Pol1. It replaces the RNA primers with DNA. All right, now we have DNA. So we're basically done here, except notice the way I've drawn this. Where the primer was, the connection between the primer and the DNA after it, there's a gap. And the reason for that is because DNA will not be added to the end of an RNA molecule. And as it turns out, the primase also doesn't connect the two. So our last step, if we get down here to 4, is that we need our DNA ligase enzyme to fill in those gaps. So that's the function of the DNA ligase. And this whole thing just proceeds down the length of the DNA molecule from both replication forks until the entire genome has been copied. And so what you'll notice is that each new DNA is going to contain one strand inherited from the parent cell. That's what's shown in red. And then the other strand has been newly synthesized. All right, so if you think back to the list of the different polymerase complex components, there was one of them that I didn't mention at all, and that's the beta clamp. So the beta clamp is not an enzyme, but it's a critical part of this enzyme complex. The beta clamp are these rings that hold the DNA to the polymerase complex. And what that does is it prevents the DNA polymerase from falling off. So if this is our DNA, and we have our replication forks down here, the entire complex, because enzymes, you know, they have to move around a bit. They have to have weak intermolecular forces holding them to their substrates. It's, you know, weak intermolecular forces are weak. It's possible that it could come off. The beta clamp sits on front, and it prevents the polymerase complex from falling off. And this is important for the reason of what's called processivity, which means that you're able to replicate DNA thousands of times without ever losing your substrate. That's very important because what would happen if the polymerase fell off the DNA? Well, if your RNA or DNA polymerase fell off the DNA, it could always rebind, but where? Where to pick up? Remember, these enzymes do not have brains. They don't remember where it was that they just fell off. They just bind and unbind blindly by intermolecular forces. And so there's no way of knowing if the polymerase fell off that it would bind in the same spot where it had just been replicating. In fact, it's very unlikely. So in order to make this process as unlikely as possible, nature has selected for the beta clamp complexes to make sure that our polymerase stays put on the DNA. Here's a list of a few YouTube animations that I think do a pretty good job of illustrating DNA replication. The first one is essentially just a recap of everything I just said. This one at the bottom, though, the last one, actually gives you an idea of what this whole complex looks like and how it works. And one of the things to notice is what happens on that lagging strand where we have to synthesize our DNA in separate little chunks. So that's an interesting video to watch. I refer you to YouTube to check out these videos. Our next topic in lecture number 20 is going to be RNA transcription. And as we're going to see, RNA transcription is very similar to DNA replication in some respects, but it has some important differences. So I'll see you guys after the next lecture or at office hours.